Welcome to this video talk, An Introduction to Anaerobic Digestion. My name is John Mullett and I work for Sustainable One World Technologies, a social enterprise. The plan for this talk is to try and achieve three things: an awareness that anaerobic digestion is a natural process, an understanding of what the process is in broad chemical terms, and an understanding of some of the key parameters. I would also like to introduce you to some of the jargon or specialist terminology that is used in this sector. I will start by talking about the possible fate of a leaf that falls off a tree. It can fall onto the forest floor and it will turn into compost, and compost heaps get hot. It indirectly releases energy and carbon dioxide via the respiration of microorganisms. It can fall into a fire and it will burn and release energy and carbon dioxide. Both of these processes need oxygen. Or it can fall into a forest pond where there is no oxygen. It will decompose anaerobically and make bubbles of pond gas, i.e. biogas. This process is widespread in nature, but an understanding of where in nature it is found does help with an appreciation of how managed digesters work. These pictures show methane being burnt as it is released from the ground in the polar areas. AD occurs over millions of hectares, albeit slowly. AD also occurs in the very hot waters found in thermal springs, such as those in volcanically active areas as Iceland is. But perhaps the best known is the cow, or rather all ruminants. Ruminants host the anaerobic digestion process in their many stomachs, and they use anaerobic digestion to break down vegetation. Without the AD microorganisms, they would be unable to derive their nutrition from the vegetation. Jargon alert. Mesophilic digestion is optimized at 38 degrees centigrade. This is because we harness in our AD plants the natural cultures of microorganisms which have evolved to live in the guts of ruminants. And so-called thermophilic digestion performs best at temperatures over 50 degrees C, temperatures similar to that found in hot springs. So the point is, humans did not create AD, we simply harness it from nature. So what is decomposition AD style? Well, using glucose as an example, you can see on this slide how the components of glucose, 6 carbons, 12 hydrogen and 1 oxygen, would be broken down into 6 molecules of either carbon dioxide or methane. Note that in this equation some additional oxygen would be required. Both of these molecules are given off as a gas, so biogas is the gas derived from this form of breakdown, and as oxygen supply is limited, there is usually more methane than carbon dioxide. The proportions vary, but the gas is usually burnable because the methane is combustible. The process that achieves this breakdown has multiple steps. These can be seen on this slide. I would want you to note that one part of the process makes acid, acetogenesis, and the latter part of the process, methane, is made during methanogenesis. The methane-making bugs do not like conditions that are too acidic. I will allude to this later in the program. The other key word is hydrolysis. During AD, a considerable degree of liquefaction occurs, i.e. solid material becomes liquid. To illustrate this, think of an apple which has been left too long. It becomes a puddle of sludge. Apparently solid organic matter turns into a liquid. I would like to go on to explore how you would take control of the AD process. There is a helpful similarity between AD and brewing. Both are completely natural processes. We find drunk wasps in nature, intoxicated with the alcohol produced by yeasts on sugar-rich foods like fruit. We can upgrade the process to a do-it-at-home one, to produce wine or beer, or even moonshine. However, you can also turn it into a major part of our lives by making it an industrial process. AD in the world of humanitarian aid is at best in the middle, but mostly on the left of this screen. It happens, but it is not managed. So what are the process parameters that matter? The processes that matter can be broadly grouped into three categories. Things that the design of the equipment or plant influence, what you put into the facility, and then how you manage the facility. I will skim through some of these as a brief guide. The photos all illustrate AD plants, a wide variety of shapes and sizes as you can see. The design of the kit or the plant will affect factors such as temperature, microbial biomass, depth and pressure and mixing. I alluded to AD in nature being found in a wide variety of habitats, cold, warm and hot. Bug cultures evolved to live in these particular natural conditions, so they have different optimum performances at different temperatures, as this graph illustrates. Most managed AD plants operate at a mesophilic temperature. If your digester is not at 38 but drops to say 30, the performance drops from 50 to 30. If the temperature drops another 10 degrees to 20 degrees C, then this figure goes further down to under 10. The performance unit on this graph is the growth of microorganisms. Temperature is absolutely key to digester performance, and those not at optimum temperatures perform well below potential. I would like to emphasize this point on the following slide. This data is taken from research work undertaken to optimize fecal sludge treatment in an industrial plant. The key point I would like to make is a 4-degree difference between 35 and 39 affects gas production by 22% and increases the breakdown of the volatile solids, i.e. the waste, by 40%. There is very little attention given to operating temperature within low technology AD, but it is perhaps the single most important process parameter. The bug culture or microbiome of the digester is what makes it work. So how do you keep them in the digester and how do you not flush them out with new waste? There are a number of strategies to do this, but most are reflected in the design of the structure. The anaerobic baffle reactor, or ABR, retains biome by having a very large surface area to volume ratio, and the bugs stick to the walls. Other options used are retention of sludge and structures within the digester to create fixed films. The pressure at the bottom of a deep digester is higher than that of a shallow one. This affects how much gas is in solution and therefore the chemistry of the process. Think of opening a bottle of beer. The dissolved gas helps the digestion process. Most managed digesters use some form of mixing to improve the process. These can be mechanical, like a propeller, gas mixing, or a multi-chambered design. In the West, all AD plants have sludge mixing to improve the process. The bugs need a balanced diet, just like us, carbohydrates and protein in an appropriate ratio. In AD jargon, this is usually termed the carbon to nitrogen or C to N ratio. Again, like us, we find it easier to digest processed food, so AD plants usually have some form of shredding or grinding to reduce particle size. A reduction in particle size by feedstock shredding is usual to make the waste more accessible to the microorganisms. We have looked at how the design of the digester affects process. We have looked at how what goes into a digester affects the process. So finally, one quick point about control of the process within the digester. My final analogy is, again, like us, the digestion process goes wrong if we have too much acid. We call it indigestion. Digester cultures crash, i.e. die off, if they are fed more than they can process. This is because the methane making processes cannot work as fast as the acid making ones. In order to manage this balance, the chemistry of the digester is monitored, so this may include pH, fatty acids and alkalinity. So, to wrap up, AD is a microbial degradation which happens when no oxygen is present. The process is natural and ubiquitous. It is widely used to process fecal and other wastes in the West. Plant design affects process management and monitoring important. Above all, it is a living culture of bugs which do the work, so look after them. Thank you for listening. My name is John Mullett and I hope it has been helpful.